Okay, you got it.
All right. Well, good morning, everyone. Hope you all had a good weekend. Um, I got the midterm exam up, but apparently I forgot to deploy it, so I did that this morning. So I'll give you guys a week from today to do the midterm. So next Monday, October 21st at 11:59 p.m., the midterm exam will be due. Let me know if you have any questions or issues with it. It seems like every year there's always some sort of problem. But I ran through it and gave a little practice. It looks like it's working okay. So I'll also open up the quizzes there for Chapter 6 and then Chapter 7 after we finish it today. So speaking of which, let's get on to Chapter 7 here. We left off talking about fruit formation, flowering and fruit formation, the sexual reproduction of plants, and I wanted to mention briefly this unique thing that some plants, some of our crops can undergo called parthenocarpy, which is the formation of the fruit without pollination or fertilization. Generally, though, since they're not fertilized, we don't have seeds being produced. So we have parthenocarpic fruits being seedless, which could be a good thing for particular crops. And there are some ways that we can actually induce this. In some cases, it is actually genetic, but we can induce it by the application of some of the plant hormones that are naturally there, such as the gibberellins and the auxins. However, not all the seedless fruits out there that we have are parthenocarpic. For instance, some seedless grapes, they're still having fertilization, but the embryo is aborting itself, so there's no viable seed being produced. In that case, it's stenospermocarpy, where it's fertilized but not actually developing into seeds. And in this case, we have to reproduce those plants through vegetative asexual propagation. We actually cause this kind of phenomenon for some plants, like the seedless watermelons, for instance. And it's really kind of a fascinating thing with the seedless watermelons. And again, it's this whole process of experimentation, how they kind of came up with this. But what they'll do is they'll be growing several varieties of watermelons, and they'll have one variety that is exposed to this chemical colchicine. And colchicine is actually a naturally occurring chemical in the crocus plant. So they extract this or synthesize colchicine, and then they put it on the plant, which induces this tetraploidy, which is basically making 2 extra sets of chromosomes. And so when we get to that point and we're creating these extra chromosomes, when the plants are cross-pollinated, and they do this in a semi-controlled environment with a regular diploid variety, because of the crossover of the chromosomes, there are extra pairs. The resulting seed becomes triploid, which induces this stenospermocarpy, where the embryos abort and they develop some seeds. I mean, like you can see in the picture here, I'm sure you guys have probably experienced this if you've eaten seedless watermelons. It will develop the white, unviable seeds in some cases, but very few of them. So it's kind of a multi-year process to get this to happen for seedless watermelons, which is pretty fascinating because we do that on a commercial scale. We're talking about the other tissues that are sometimes involved in fruit formation. We refer to those as accessory tissues. The actual true fruit, though, is just the enlarged ovary of the plant and kind of the pulp associated with that. However, we will have some accessory tissues that are involved in the fruit's formation and often still attached to the fruit. That's just a difference in semantics there, though, how you refer to it. In many of our crops and in many plants in nature, only a small percentage of the flowers actually develop into fruits. And this is just kind of a natural fail-safe for plants where they produce more flowers and in some cases more fruits than they actually need to try to become more survivable and pass along their genetics easier. So here's just an example of some of the accessory tissues in this case in a lettuce fruit. We don't really think of it that much as a fruit because it's a dry seed, but technically it is a mature fruit. So we have fertilization, pollination, and a process of it developing from that fertilized egg into a mature fruit. And this becomes really important when we're trying to save seed to regrow it. We want the seed to be as mature as possible. So we look at certain harvest indices to make sure that they are actually viable. And in general, we want darker seed, more firm seed that is extremely dry. There are also a lot of different plant hormones that can be involved in the fruit being set, and a lot of that science is still not well understood on how that actually happens. However, we can, in some cases, apply these exogenous hormones, such as auxin, to replace pollination or fertilization. And we might have to do that in some cases, like in growing in greenhouses or controlled environments where we don't have natural pollination occurring. We also use some of these hormones to not only induce, but also change the way that the fruits look in some cases. For instances, commercially, we're always applying gibberellins to grapes, for instance, to make them larger. So when we're trying to produce fruits on a crop level, we want this optimal level of fruit setting, and we have to have the optimal conditions for that. So if we have too low of a fruit set, of course, it can kind of eat into the profit margins of the crop. However, on the other end, too heavy of a fruit set can damage the plants, and also the more fruits you have, the smaller, and in some cases less quality of the fruits that you'll have as well. So one way to overcome excessive fruit set is to actually do some thinning of the fruits at usually a very early stage, after fertilization, after pollination, but before that energy is put into developing those fruits and making them larger. Some plants are self-thinning, and you'll see that a lot of times, like in peach orchards, for instance. They produce a lot more flowers than they need, a lot more fruits, but then they end up aborting, sometimes up to half of them and drop them. So self-thinning is actually a desirable trait to look at in some varieties, because it means the fruit that is remaining is going to be larger. We also have issues with temperature, light intensity, low soil moisture that can adversely affect the setting of fruits. Ideally, we want the optimal conditions, so the right temperature, not too low, not too high, the right amount of light, and of course the right amount of soil moisture, because those fruits are, the majority of them is water. So using some of these hormones in actually growing the fruit as well as just getting fruit to set. One of the best examples of that is using gibberellin on, and they use this on many different types, varieties of grapes, but the classic example is the Thompson seedless grape clusters. And so when those fruits are beginning to develop, they apply this gibberellin, which causes the fruits to continue to grow. And you've probably seen, small grapes out there at farmers markets where they haven't applied gibberellin, but for the vast majority in the grocery stores, almost all table grapes are treated with this hormone in order to get them to increase the size that the fruit makes and make a larger cluster, which leads to greater profits, of course. So when we're looking at the stages of the development of the flowering and then of the fruiting, we have cell division occurring. And once we have that fertilization, it's pretty rapid. That lettuce example, we're looking at like 10 days between, pollination, fertilization, and the development of a viable seed. So we have really intense cell division, and along with that, we have fruits increasing in size. And there are two basic patterns of this growth. Generally, where there will be a slow start initially and then a rapid size increase. And then once they kind of get to that, close to that maximum size, they will decrease and then just develop the sugars and the chemistry of the fruit changes. And then there's kind of in other crops, and this is mostly in the kind of linear flow, we have the oranges, apples, almonds, and then in other plants in the same family, it will have a rapid increase in fruit size, followed by a slow period where they're not really increasing, but the chemistry, the chemicals in the fruits are changing, and then they increase in size again before they mature. There's also all kinds of different, biochemical changes that are happening in the seed as well. So in this case, looking at stone fruits, initially the fruit develops, and when they have that lag period, this biochemical change is taking place in the actual seed itself. So you can see here we've got kind of just the accessory tissues with a smaller endosperm. The endosperm begins to get larger and kind of replaces those secondary tissues. And then the embryo begins to grow and takes up most of the tissue of that seed. And then the fruit begins to grow again. One thing that we need to think about and look at is how some fruits will ripen on the tree. Some of them will continue to ripen off of the plant rather than just on the plant. And these are called climacteric fruits. And generally what we do commercially in the cases of these climacteric fruits is actually gas them with this, it's a natural plant hormone, ethylene, that these fruits are naturally releasing. But we, in some cases, on the actual fruit trucks on the way to, their final destination in the grocery store or at the market, they're gassing them with ethylene. In some cases, at the grocery stores, they have rooms where they're gassing them with this natural plant-based gas in order to get them to continue to ripen. So apples are really good at producing this gas and releasing it, and that's why you don't want to store your apples with certain plants, because ethylene accelerates the ripening, or in some cases also the decay of plants. So if you have, say, flowers in the same room as something that's releasing these ethylene gases, then it will cause the flowers to continue to develop and drop their petals drop the showy parts. Some of them will begin to rot too. If you have apples and onions together, the apples will cause the onions to sprout. So apples, apricots, bananas, and tomatoes are just some examples. And especially on the banana and tomato side, we actually are picking these fruits when they're still green in many cases, and then using that ethylene to accelerate the ripening. So we don't actually, in that case, we're not getting the same flavor profiles as we get if they just naturally ripen on the plant. So we see like the greenhouse vine ripened tomatoes in the store. There are different varieties that they're picking on the vine. They're not gassing them with ethylene, so they taste better. But the saw fruits are more shippable when they're green, so that's the big reason why we're doing this. So we can also hasten them by treating them before or after harvest with the ethylene as well. So kind of back to the lifespan of plants, we kind of mentioned, some plants can be extremely old. All kinds of different lifespans in our crops. So some that range from just a few months in age, one growing season till they, go through maturation and develop seed and then die in our annual plants, to, in some cases, thousands of years. So coast redwoods, for example, some of them are known to be over 3,000 years old. Olive trees in the Mediterranean, in some cases over 2,000 years old, and we're in cultivation. They were planted as a crop. And even though they have this great lifespan, does that necessarily mean that they're going to be productive for their entire lifespan? Probably not. I mean, there's going to be kind of a cycle there as well, where they're going to have kind of their prime period in between kind of juvenility and maturity, where they're producing the most of the crop. So when a plant reaches maturity and then reaches, starts to get close to the end of its life cycle, we have senescence, which is this irreversible change that leads to the breakdown of cells and tissues, and then eventually the death of the plant. So we'll start to see, considerable decline that is very evident near the end of their life cycle. In annual plants, we see that along with the environmental cues. So once we start to get those low temperatures, they're going to senesce and die. We can see this in plant populations or in individual plants, or just in specific organs of plants as well. And we see this in many different, kind of culminate in different ways in different plants. So in the annuals, the whole plant dies back, root system dies. In the herbaceous perennials, the tops of the plants die back to the ground at the end of the growing season. But then there's energy that survives there in the roots. The roots continue to survive and live for many years. And then the crown of the plant will expand and a new shoot will grow the following growing season, once those environmental cues are reached. In deciduous woody perennials, we have the leaves actually senescing and dying and fall off every year. But again, it's triggering dormancy in the buds. So we have shoots that remain alive as well as the root systems. And of course, in evergreens, they slow down their growth and they will drop some of their leaves, but the shoots and roots remain alive throughout the year. So can we extend the life of a plant? This is actually something that we look at in the bonsai world. We visited a bonsai grower who used to maintain our national collections of bonsai. When we started to develop relationships with a lot of Asian countries, they actually gifted us all these bonsai, which were, hundreds, in some cases, thousands of years old. And these are, vegetatively propagated clones in many cases, and they are actually extending the life of that plant. And so this bonsai guy that we were visiting was basically saying that, these trees that he's taking care of are, thousands of years old, because he's taken that, cutting from it and extended its life. And theoretically, we could do that kind of indefinitely. And I don't know how you all kind of view that, but, is a clone the same, life form, or is it a different life form? It has the same genetics. But I mean, we have some plants out there that we've been growing the same varieties for hundreds of years, and they have to be asexually propagated, like apple trees, for instance, through grafting. So we're, in a way, extending the life of that plant. So one example is the winter pearmain apple that was cultivated in England. They have records going back as far as 1200 AD. So it's been in cultivation almost 1,000 years. Kind of crazy. The black Corinth grape has been grown in Greece for thousands of years. It's fascinating. We also can have senescence not just due to the life cycle of the plant, but caused by environmental stresses and or other life forms, organisms such as the pests, pathogens, and parasites that attack them can trigger senescence. And when we have this senescence beginning to occur, we have the actual chemicals in the plant begin to degrade. And important to note, the DNA and the RNA of the plants are sometimes the first to break apart, so the plant can no longer reproduce at that point. Senescence, however, is not entirely just degradation of the plant. In some cases, you only get particular messenger RNAs and proteins that are synthesized when the plant begins to senesce. So that could be an important thing to look at in particular crops. In some cases, we actually try to hasten that senescence. And we look at that as the transfer of stored nutrients to the reproductive parts of the plants. We can actually delay it in some cases by just picking or pinching of flowers, or the removal of fruit before the seeds start to form. So basil is a classic example of that. We want to continually harvest the leaves, so we're not going to let it get to the part where it starts to senesce. So we remove those flowers to keep it vegetative. A lot of plants that we might do that with to try to get them to produce vegetatively as much as possible before they get to reproductive capability. This whole cycle of life in plants is similar to the same as what we have in animals, and that it is controlled by hormones, these chemicals that correspond to the maturation and then the senescence of the plant. And there's a distinction here between a plant hormone versus a plant growth regulator. The hormone itself is actually the natural substance that is produced by the plant and controls the plant's activities. Plant growth regulators are sometimes are plant hormones, and they can either be natural, found and extracted from the plant, or synthesized in a laboratory. And they are applied, to a plant to trigger these different parts, developmental patterns in the plants. So we have 5 traditionally recognized groups of the natural plant hormones, and they are the auxins, the gibberellins, cytokinins, ethylene, and abscisic acid. And on this table here, they're taking a look at the naturally occurring hormones, as well as the synthesized forms and how the chemical structures can be somewhat different in these different formulas. Although note that like ethylene, for instance, is pretty much the same. Abscisic acid is the same. Some of them are altered when we synthesize them because we can't reproduce that actual natural mechanism that they're creating them through. We've also recently added hormones and hormone classes that we're still not great at getting a complete understanding of. We have the brassinoleads, salicylic acid, and jasminates are recently added hormones or hormone classes. So let's talk about each of these hormones. First, we have the auxins, which are operating at a cellular level, and they affect different activities in the plants, but they originate in the meristems of the plant. So where these tissues are basically being differentiated to be either like a stem tissue, a leaf, flower, etc. So auxins are naturally found in the shoot tip of each branch and generally at the highest point of a plant as well, where we have these meristems. And auxins are what causes this phenomenon known as apical dominance, where you have one central leader in the plant, the highest growing point, and everything else is kind of below that point. And this is due to a blockage of the growth of lateral buds by the presence of the terminal buds, where these meristems are and where the auxins are stored. One of the most widespread auxins we have naturally in plants is indole acetic acid, and it's one that naturally occurs and we've extracted from plants, but we've also begun to synthesize different forms of it in order to create rooting in plants. So we use it as a rooting hormone. It kind of comes in a powdered form. So in creating cuttings, we actually will dip it in this powdered or liquid form of this auxin hormone to develop adventitious roots. We can also use it in weed control, inhibiting stem sprouting, and also in the use of tissue culture types of propagation, where we're reproducing plants in like lab conditions. The next class is the gibberellins, which basically stimulate cell division, elongation, and in some cases both, and also control the secretion of particular enzymes. Gibberellins are also involved in flower initiation and whether the plant is becomes a male or female in its flower. So fruit set as well as fruit growth and ripening, it can be controlled by gibberellins. Different species have different effects of gibberellins. In some cases, if we actually apply gibberellic acid, then we can get dwarf cultivars of particular crops to grow to normal heights, which basically just is indicating to us that the dwarf plants lacked the normal level of gibberellin. So here's just an example of that. In the left container there, we've got an untreated genetically dwarf corn. And then in the center, we have a non-dwarf corn that has been sprayed with gibberellin. As you can see, the leaves are developing much longer than natural. Now on the right, we've got that genetic dwarf that has been sprayed with gibberellin, which is basically causing it to break out of that kind of dwarf phenomenon. Gibberellins are also important because they are involved in overcoming dormancy in seeds and buds. So when we try to get a seed to germinate, it's actually naturally, kind of catalyzing that gibberellin that is produced in the embryo and moving it into the seed coat or to the seed coat so that it breaks the dormancy of the plant, which basically just causes the genetic changes, utilizing those enzymes and synthesizing the proteins that then become available for the embryo to actually grow and develop. Gibberellins were first discovered in 1926 when these Japanese researchers were studying a rice disease that was caused by a fungus. They found that the plants that are infected with the fungus grew excessively and abnormally, and then they extracted some of these chemicals from the fungus and applied it to non-infected plants, which stimulated that same abnormal growth, which later on they, kind of synthesized that chemical, and they figured out what it was, and they named it gibberellin from that Gibberella fungus. Here's the example I was talking about in the grape example. So on the left, we've got an untreated cluster of grapes, and on the two on the far right here, we're looking at them being sprayed with different levels of gibberellins. And that's basically just causing the tissues to continue to expand and the grapes to grow larger. Note, though, that we also have this stem girdling process in the second one from the left, and that's kind of doing the same thing because it's inhibiting different hormones from actually getting to that. So it's limiting the auxins and actually increasing the gibberellins, which is causing them to grow larger too. So here's another example in grape seeds. So on the left, we've got no gibberellic acid, and then we have increasing amounts over here, which cause greater germination of the seeds. So they are very important and powerful for particular types of production, but there aren't a ton of different uses for them. So, I mean, we can promote male flowers in cucumbers. We can use them to overcome the cold requirement for stratification or kind of vernalization in some plants. And in some cases, we use them in ornamental plants, such as poinsettias and geraniums, to get them to grow more into a tree form, because the gibberellins are involved in that stem elongation, which just makes a different, ornamental, more aesthetically pleasing form of that plant. So here's an example of that, where we've applied gibberellins and removed the side shoots to kind of make them a more attractive kind of tree form rather than just the more bushy kind of natural plant. The next class of hormones is the cytokinins, and their primary function is just to promote cell division, but they also play a role in some of that cell growth and development, the enlargement of the cells, differentiating tissues as well, also to a certain extent in dormancy and in the retardation of leaf senescence. So the big deal is that cytokinins are interacting with auxins to influence the differentiation of the different plant parts in the meristems. So, I mean, the big thing here is whether we're getting shoot or root growth, and it's the levels of auxins and cytokinins and the ratios of cytokinin to auxin that is really controlling whether a plant is producing a new shoot or a new root. There are several natural and synthetic cytokinins out there, over 100 now that are known, and they occur in many different plant tissues. So here's just an example of the effects of cytokinin and auxin, and in this case we're looking at kind of a tissue culture. So on the left we have a piece of the plant. Tissue culture is kind of cool because you can just use theoretically just one cell of the plant and apply the different hormones to get them to grow shoots or roots. So in B here, we've got the application of cytokinin, which causes shoot production, but we don't have any root formation. In C, we're using auxin to trigger root formation, but they're not developing buds and eventually shoots. In D here, we've got cytokinin and auxin kind of at an even level here, which is, kind of beginning the callus growth, which is going to allow both shoots and roots to develop, but we don't have it at the proper ratio to get both yet. So they also indirectly increase enzyme activity and increase the DNA produced in some of the tissues of the plants. We say, at least in the book, they're saying that there is no important agricultural uses that have been developed for the cytokinins alone, but just in the propagation of the plants, it's very important. So in getting them to develop shoots and roots, and it's very important in plant tissue culture. So ethylene, we already started to talk about, this natural gas that is produced by plants, found as early as 1924 when they found that it induced fruit ripening. And they found in the 30s that it was a natural plant product and that it was, being developed by the ripening of the plants. It naturally occurs as a gas and diffuses readily throughout the plant and outside of the plant as well, and moves in many ways very similar to carbon dioxide. And it can actually exert influence on plants in very small quantities. So we actually use a synthetic form of ethylene in the chemical ethifon, which is applied to plant tissues to produce more ethylene. So again, we actually look at that and using it as an agricultural spray to cause the release of ethylene to accelerate ripening in plants. So here are the commercial applications for ethylene. First, just the ripening of fruit, like we mentioned, the initiating of flowers in some plants, and you have to be careful with, the application of particular hormones because they have different responses, not even just in different species of plants, but in some cases different varieties. It can be used to change the sex expression of flowers if used before the flowers open. We use it for the degreening of oranges, lemons, and grapefruit. You can use it to aid harvest in some cases because it's continuing to cause maturation and might, allow the fruits to naturally drop from the plant. We also use it to regulate growth. And it can also harm plants. So it can cause unwanted leaf drop and can hasten the process of senescence of most flowers. Also can cause flower bud abortion of bulbs during shipment. We can also get ethylene being produced by gas or oil-fired heaters as a byproduct if they're not vented adequately as well. All right, the next class here we have abscisic acid, which is basically what causes the abscission, the dropping of deciduous leaves in plants. And we have two major roles in the life cycle of the plant. First, the regulation of the processes in seed development, and then just as a hormone that initiates responses to cold and water stresses. So, in the drought, we start to see plants turning brown and dropping off. That's just because they're initiating that abscisic acid as a response to that drought. And it's kind of a survival mechanism. So they're doing that so they're not putting, because they don't have water, they're not putting more energy into moving water into the leaves. Instead, they're just dropping the leaves so they can put more energy in developing the buds for next year. Abscisic acid is synthesized in both leaves and roots. And currently, there are no large commercial uses of abscisic acid in crop production. However, it's important just to know what role it has in the life cycle of plants. So the other hormone classes that we mentioned, the brassinolides are steroids, which are actually very similar in structure to animal hormones such as estrogen and testosterone. It's still not clear exactly what they do in plants, but in some cases they're actually extracting them from plants. For instance, birth control products, they extract a similar chemical and then synthesize it in progesterone from hops flowers in order to create these birth control products. Salicylic acid is what the natural component of aspirin. Before they synthesized it, they were actually deriving it. Native Americans, we mentioned that back when we were talking about the ethnobotany. They were using it as a natural painkiller, and it can be an important component of the plant's responses to pathogen attack. The jasminates are a group of compounds that, again, kind of similar to salicylic acid, are a defense hormone to trigger changes in the chemical composition of leaves to make them less palatable from like herbivore attack, pests that are feeding on the leaves. And cysteamine is one of the newer hormones that's been classified. It's unique because it's the only one that they know to be a peptide and is composed of a bunch of different amino acids. And it is produced in wounded tissues and then is transported where it's basically being used to induce defensive genes. And right now, we don't really have commercial uses for the application of these hormones, but with that plant science and technology rapidly developing, we may one day find uses for these in crop production. We also have some growth retardants that are used and have been important in some commercial uses. The big thing is to obtain more compact dwarf type, more aesthetically pleasing plants in ornamental crop production. So there's slowing cell division and elongation, which keeps plants more bushy. It will also cause plants to have darker, more attractive foliage and more flowers than the untreated plants. And most of these are acting just to block gibberellin synthesis, so they're reducing that stem elongation, making it look prettier. So here's just a few of the better known synthetic growth retardants that we use, both the chemicals here as well as their this is like the trade chemical itself, or the common name of the chemical, followed by the scientific name of the chemical, and then the products that you find them in. So like B9, Psychocell, paclobutrazol, used in bonsai, very important in the ornamental crop production world. And that's it. So that takes care of chapter 7. And we'll get those quizzes up for you guys on chapter 6 and 7 here in just a few days. Open those up for you. So if you have any questions, comments, or concerns, just let me know. Otherwise, have a great Monday.
Okay.